extremely excited to be here today because what we have as a panel today are exemplary scientists from the country and from USA. And I really, really look forward to hearing from them all about genomics and proteomics. So first we have Professor Kannan, who will be speaking on Frederick Sanger. So this series is about on scientists whose contributions have made seminal impact in the field of biology, so much that has improved the lives of humanity. And needless to say, Dr. Frederick Sanger, two-time Nobel laureate, is one amongst them. Professor Kannan will be speaking on contribution to the primary structure of proteins and the work of Dr. Frederick Sanger. Professor Kannan is a man of several hats, having worked both in academia and industry. But I think at the core, he is a protein biochemist and has contributed immensely towards understanding of protein biochemistry. And a seminal contribution from him has come from the evidence of extracellular ubiquitin, which plays an important role in hematopoietic stem cells. Professor Kannan was the first dean and professor at the University School of Biotechnology, Guru Gobind Singh Indraprastha University, and that's from where I know him, from the first batch of biotech students there. And from there, he moved to as the vice chancellor of Nagaland University, and then he was a vice chancellor at the Nagaland University, and presently he's the founder of KKBiotech. So Kannan sir, over to you. Please tell us more about Dr. Frederick Sanger and his contribution towards understanding primary structure of proteins.
Yeah. Thank you, Bhavana, for a very, very kind introduction. And it's always a pleasure to see all of you. And let me just start sharing my slide.
Sure. I'll just take 1 minute to tell the audience that please feel free to put your questions in the chat box. I'll probably take them all up towards the end. Of course, you can always also ask questions towards the end after Kannan sir's talk.
Yeah, welcome to all of you for the second monthly lecture series on scientists who lived so that humanity lives longer and healthier. Last month on the same day, we spoke about Yellaprakada Subbarao and how many lives have been saved. And today we are going to talk about Dr. Frederick Sanger, whose contribution will continue to make an impact in the coming years, even though he is no more with us. But I think his contributions will remain. Today I'm going to talk about evolution of protein sequencing. And today, I'll be talking essentially about how Sanger sequenced insulin. So that is a pure protein.
Sir, one sec. Could you just make it full screen so that it's bigger for people
yeah,
to
sorry,
see?
sir. I'm so sorry.
Yeah, no problem. Thanks.
Yes. Yeah. So, when he was fortunate that thanks to Eli Lilly, he got 300 milligrams of insulin. So he could play around with it and arrive at the structure. And we have other extreme today. We are going to hear Dr. Swaminathan, Jagannath Swaminathan, who will talk about his exciting work on single cell protein. As you can see, I've divided the lecture into various parts. One is 1900, then 1950s, what happened, 1970s, what happened, 2003 onwards, what has happened, and where are we now. So you will see a certain flow of thoughts which comes into the whole thing. Just before I begin, this KKBiotech was essentially started because when I've been visiting professors or holding the chair of professors, I found that suddenly children are a bit, worried. Have they done the wise thing to join biotechnology? And looking at many of them, I felt, how come this is happening? Because uh, when I was a professor in the university, I saw there was a lot of enthusiasm to join that. I remember Guru Gobind Singh Indraprastha University, we had, for every seat, we had nearly 300 applicants at one point of time. So yeah, there was a lot of excitement. So I'm wondering what happened. Anyway, I said, what can I do for it? There's no point talking about, what's not. So I said, our idea essentially is to inspire, ideate, and implement. So that's what the vision is. And you'll be happy to know that we have also started the internship program, and and a lot of students are taking interest in this thing, and we spend about every day about 3 hours with them, when the workshop really starts. So we have begun somewhere, and basically we are trying to cater to catching up the young ones who are in 11, 12, because I had the pleasure of introducing biotechnology in schools under CBSE, and Bhavana was one of the people who helped us to, edit the book, correct the English, and all that. So I remember those days. That is way back in 2002, and then we, bachelor's program, master's program, PhD, postdocs, because I'm suddenly finding postdocs are also a bit confused, and of course professors and teachers, and I can quite appreciate what the problem is. So we thought we'll focus on education, professional development, because we have to give confidence to the youngsters that, look, it is you who has to create an opportunity doesn't come your way. So one of the things, I'm helping some people to frame research proposals. We are trying to push some people into entrepreneurship. There are some interesting models I'm trying to work out on. Then help in research and development, and latest one we have added is internship for students. If you want to know more, please do visit our website. So objective of today's talk is, how does one identify a problem? How do you do the molecular identification? What are the techniques to detect proteins? And transitioning into fluorescent methods for detection. So this will be the general flow of the talk today. And so, as you know, diabetes has been the bane of our society. And before 1921, the number of, if a child has juvenile diabetes, suddenly the childhood version was deadly, striking suddenly, and the child will be no more. So it was pathetic to watch this situation, and uh, certainly the people knew about diabetes, especially Thomas Willis had talked about it way back in 1675, that it is sweet and etc. And he said, because they are not able to process glucose in the body. So, and today India is supposed to be having the largest number of diabetes, and people say it is more than 50 million, which is quite scary, because in a population of 1.3 billion, 50 million is quite a significant number. So, how was this identified? Because we are now going back to 1921, okay, and a young doctor, Banting, along with his junior called Best. Banting and Best is a classical one. They did the following experiments. They ligated the pancreas, so that the pancreatic ducts were ligated with silk thread, and then what they made sure that, and they also did a beautiful experiment of making sure all the enzymes are released, so that the the active principle will not be destroyed. As you know, even today when we try to isolate any enzyme or any protein from any source, we always put proteolytic inhibitors, because if the proteolytic inhibitors are there, they may destroy the active principle. So those days, because they were physiologists, so they knew the physiology, what is happening in diabetes. So what they did, that it was already known that islet cells of the Langerhans has a role to do with diabetes, but they didn't know how they are, what is the role, etc. They didn't know. So Banting and Best were the first one who sort of ligated the duct of it and then made sure all the proteolytic enzymes are flushed out, and then the atrophied one, as you can see on your slide. Okay, here you can see the atrophied one. So they took the atrophied one and then took the extract out of that. Then what they did is they simulated what is called an animal model of diabetes by this thing. So this animal has become diabetic dogs, as you can see. Right here is one. There is another one. They gave that extract and suddenly the sugar levels came down and the dog was active. Whereas the second one, where they did not give, it remained sluggish. So the glucose level hadn't come down. So this was a very dramatic experiment. They said, okay, and now we can bring down the glucose level. So 1921, there was a mad rush of people traveling from USA, etc., to Toronto in Canada to get treatment. And they were willing to pay any amount of money. That was the time when the company Eli Lilly came forward. And as you know, no patent was taken for this. In fact, it was given to Toronto University, the discovery, for $1. And that's why Eli Lilly went in a big way. And Eli Lilly has been the first player in bringing out the therapeutic treatment for this thing. Mind you, we are talking it only as a therapeutic treatment, but there's not a cure for diabetes. Please remember that. So it's not a cure, but I can make people live longer. Earlier, the children used to die within 4 or 5 years. So that one disappeared and people started, having a healthy life. That's why the title of our beginning, that people who lived longer thanks to some people. Okay. So it was a big news item in 1921. They didn't know what this was. They didn't even know it is called insulin. They said some factory is there. If you inject that, the glucose level comes down. From 1921, the structure finally came out in 1951, thanks to Frederick Sanger, who put all his efforts into it and arrived at this structure. So physiological function to structure took us 30 years. So that is what the Sanger started. So Sanger picked up this problem of insulin, how to solve the structure, and that is why we are going to discuss in great detail about what happened. Now, I just want to take you that at that time, people don't know what was a protein. There was a lot of confusion. There was some confusion. They said it doesn't have a chemical structure. Some of them said it's a colloid. For example, one of the classic examples is, as you know, the milk is white because there are colloidal-like particles, what we call as micelles, which scatter light, and that's why you see the milk as white. So people believe that it is just clump of several things, probably, and they didn't believe it had a structure because nobody could crystallize them. Because one of the fundamental rules in organic chemistry is that anything, if it is chemical, it should be crystallizable. And as you know, crystallizing a protein is a humongous task. It took us very many years to understand that. But one thing was very clear between in the early 1900s till about 1940s, few things people learned. Earlier, people used to do carbon, hydrogen, nitrogen estimation, but people discovered that if you treat it with hydrochloric acid, boil it 100 degrees for 24 hours, any protein, they will break up into the constituent amino acids, which are the building blocks of the proteins or peptides. So people started looking for amino acids what are present rather than looking for carbon, hydrogen, nitrogen, etc. Number two, they also discovered when they hydrolyzed protein, they found in general there are 20 possible amino acids. And these amino acids were unique in the sense that they had in the carboxylic acid, the carboxyl group, the next carbon always had an amino group. Okay, and interestingly, even though it had carbon, hydrogen, nitrogen, etc., these molecules, what is called amino acids, did not truly behave like an organic compound. For example, when we say organic compound, they should be soluble in organic solvents. these were not, they were soluble in water. Okay, or at the most you put some 10% isopropanol and it will dissolve. So it had a high melting point, high boiling point. So all this, so a lot of times people wondered whether to call it organic or inorganic, because it was a very confusion, which is quite understandable. Another most interesting discovery which happened was that this alpha amino alpha carboxylic acid can be stained with a reagent called ninhydrin, and it will give a rumen's purple. Only exceptional amino acid they discovered was proline, which is an amino acid, and when it reacted with ninhydrin, it gave a yellowish brown color. Okay, so so the ninhydrin became a very powerful way that if you take any substance, whatever pure form or semi-pure form, you can say protein is there. And there are a lot of models how these are linked, etc. And one of the models proposed was that there could be a peptide bond, but there was no evidence whatsoever. Number three, what you should also understand that these amino acids are optically active. And they found that all the amino acids which we find in proteins are L amino acids, capital L. Okay, that is, they are optically active. Whereas if you look at the sugars, they are all D sugars, what we find. So stereochemistry wise, all the proteins what we find in nature are all L amino acid. Few exceptions are there, like bacterial cell wall, which uses D-alanine to form the cell wall, so that our proteolytic enzymes cannot detect them. Okay, the only structure which was solved at that time was a cyclic peptide known as gramacidin S, which which was the first one. It was a cyclic peptide. It was also a very interesting one, which was solved, and this work actually got the Nobel Prize also in 1952. And so these are the status that they are probably linked via peptide bond, but there was no proof whatsoever. So one of the very popular one which was developed was by Martin and Sinch, who had shown that you could separate amino acids by partition chromatography. So paper chromatography became popular, and Sanger in all his work has used only paper chromatography to separate the amino acids or peptides or whatever it is. So normally these amino acids are not visible in a paper until you, as you see on the right hand side, if you spray it with ninhydrin, then it suddenly shows up color. And so this was those days when we had to separate the peptides. We only use these papers. We draw a full line of that mixture of amino acids, run the chromatogram. One of the very popular solvents we use is butanol acetic acid water, because you must remember this fact that whenever we do chromatography, you don't need salt or buffer or anything you cannot use. Because the advantage butanol acetic acid, all of them will evaporate away if you heat it to 100 degrees or whatever the temperature you want, or leave it in the air, it will dry up. So because you would like to isolate it without any salt content, because it's very messy to handle salt being present in the peptides, etc. So this was a very popular. So you draw a one and you take, you cut one small strip out of that, preparative one. You do spray it with ninhydrin. You know where the peptides are. So you mark those positions and then elute it out. Okay, just like just elutionist just put some pipette. Have a test tube in with that paper is sort of this thing, just like you put a funnel and then just pour the solvent slowly, thing will drip and all the peptides will be eluted. So you will see this was the method used by Martin and Sinch. This was the method used by Frederick Sanger also. So I just showed this to show how ninhydrin helps you to locate where the peptides are in the in the paper chromatogram from the mixture. And before this method was available, that is 5.7 normal hydrochloric acid, they used to say, look, the molecular formula is there are 257 carbon, 383 hydrogen, 65 nitrogen, etc. but that doesn't say anything. Then, thanks to this hydrochloric acid method, they discovered that there are in insulin about 18 amino acids present. Tryptophan is absent. Okay, so it is very interesting. And acid hydrolysis will break any amide bond or peptide bond. So asparagine, which is an amide amino aspartic acid amino derivative, or glutamine, they all will this thing, and cysteine gets modified to cystic acid. Okay, so but then people knew the chemistry of it, so there was no problem. If you want to see tryptophan, then we do what is called an alkaline hydrolysis and try to find out where is it. Now, normally we always estimate a protein by making an absorption at 280 degrees in a spectrophotometer, and generally the thumb rule is that 1 OD means 1 milligram per ml. This is the thumb rule, all of us used to use it. Now, as you can see, insulin doesn't have a tryptophan. So how do you estimate how much of protein is there? So these are all the interesting questions. This is not the place to discuss, but how Sanger went about doing it. And when Sanger started the work, nobody knew what was the correct molecular weight of insulin. So he had the guts in him to start this work without even knowing what the molecular weight was. So just to give you a summary before I explain each one of them. So his contribution between 1944 to 1958 has been developing a method using a reagent called fluorodinitrobenzene, which when attached to any amino acid present in a peptide or a protein, and when you hydrolyze it, this group which is attached, which has reacted with the amino group, doesn't fall off. The peptide bond is broken. So you do a paper chromatogram and you will see the yellow color, and people had studied the RF values, what you call how far each amino acid moves. And in fact, the person who did all this work is another person who became famous in the immunology field called Rodney Porter, who worked with Sanger and characterized the RF values of each of these amino acids. Okay, and he's from by this method, when they treated insulin with FDNB and then hydrolyzed it with hydrochloric acid and looked for they were surprised to find there were only 2 amino acids present. So they said probably it is made up of 2 chains, because they only saw 2 groups. So that is why they demonstrate that insulin is made up of 2 chains. Okay, because they did nothing, just took the protein, treated with FDNB, and they saw that. Now it is very interesting the fluoro compound was used, because earlier Sanger wasted a lot of time using chloro compounds, but it was not very successful. So at that time, a lot of people were, it was a war time, as you know, in the 1940s. So people were interested in fluoro compound. So a lot of labs, even in Cambridge, chemists were making various fluoro compound for war purposes. So this compound was readily available, and the head of the department, Professor Chibnall, said, "Why don't you try with FDNB?" So he got it from the chemistry department, and Sanger was very amazed to see that it works like a charm. Okay, so so they identified these D amino acids, and they discovered, as you will see, one of them was glycine and another was phenylalanine, which was at the amino terminus. Okay, then the biggest challenge was, how do I separate these 2 chains? So they discovered that you need they are linked together by a disulfide bond. So he discovered this reagent called performic acid, which could break these 2 bonds, which we will see soon. Then he separated on paper both the chains and labeled them as A and B, depending upon what the N terminus was. Even though the B chain was longer than A chain, it was much easier to handle. So one of his first students, whom you will see soon, worked on the B chain, and I will tell you that was the first time enzymes were used to create peptides. Okay, and there's a story to it, which you will see when you read Sanger's DNA sequencing, how cleverly he used the enzyme for his work. Okay, so paper chromatography was the ultimate one. You separate it, you elute it, characterize it, do the amino acid, everything they did with the paper chromatography. Okay, and ultimately, it was a jigsaw puzzle, and they started doing it, and I'll explain to you how one does it. Okay. And lastly, I will talk about protein fingerprinting, because if you were to look for the same protein, say in this case, say insulin from cow, pig, sheep, human, you can simply, when you do the fingerprinting, which I'll show you in a few minutes, you will find that there are very few amino acid changes. Many places there's only one change. So the insulin produced in all, whether it's a cow or a buffalo or a human being, the number of amino acid changes are hardly one or two. So from the fingerprint, most of them match except for a one peptide which will be varying. Okay. And he also attempted to do sequencing with radioactive amino acids, which was a miserable failure for him. But he used this again when it came to DNA sequencing very cleverly. Okay, so we'll discuss this soon. Okay, so for the first time, he established how amino acids are arranged in a specific sequence in insulin, and later they found in all protein amino acids are arranged in a particular fashion. So that was the thing, and they also established, and which you will see when I talk about sickle cell disease. There is one slide that what happened in sickle disease, hemoglobin, one single amino acid change from valine to glutamic acid causes what is called the sickle cell disease. So all these things could be suddenly done. In fact, nowadays you hear about COVID, the new variant. What we are seeing is in the protein, the S protein, so and so amino acid has changed. That's what essence. So even today we are talking the same story which was being talked at that time. Okay. So just to quickly take you through this. So fluorodinitrobenzene chemistry is given. Don't worry too much. So it is done at about pH about 8 pH, okay, because that's the only time amino group is available. So NH2 can react with this one and produce called FDNB, and then when you hydrolyze it for a 6-normal hydrochloric acid, rest of the bonds break, but the label remains on the amino terminus. So that is the interesting aspect of the method. Okay. And as I said, it is done. I just want to say a few minutes. So when the partition chromatography was developed by Martin and Singh, they found that, in paper, I told you they use what is called butanol acetic acid water. So what happened? The water remains stuck to the paper, and the butanol doesn't stick and it keeps on moving. So the amino acids are getting partitioned between the water which is absorbed on the paper and the butanol which is keeping on moving. So depending upon which is more soluble in butanol will move ahead and keep on moving with the butanol layer, and which is more soluble in water will remain behind. And that is exactly what the separation is. Mind you, I told you in the beginning that Sanger had used 300 milligrams. Then his own colleague who was with him in Cambridge and who moved over with him to the Laboratory of Molecular Biology in Cambridge came across this. He was working on one of the enzyme called chymotrypsin, and he wanted to use this compound to see the active site of chymotrypsin. But what he found was that this one goes and sticks to the amino terminus. And by this discovery, he went to Sanger, look, I want to use this and develop it like FDNB, what you developed. And this was 1,000 times more sensitive. So instead of 300 milligrams of protein, you could hardly start with 1 or 2 milligrams, and that is the thing. And later people developed what is called a polyamide sheet. So just like you look at the star center, which is with star, we run some standard, we know which are the position. By an experience, you will know this is this amino acid, next is this one, etc. You can do it. So it became a very all the sheets are hardly 2 centimeter by 2 centimeter. You must also appreciate that people have sequenced, like Hartley himself sequenced the, chymotrypsin, and single-handedly he did, because you could separate all the peptides and subjected sequencing, you find out the N-terminus. By that time, the Edmond degradation was available. So one amino acid could be removed at a time from the amino terminal, find the new amino acid which is released, test it with densyl chloride or FDNB, whatever you want to, depending upon how much quantity you have, and you will see this. In fact, I remember I used this fluorescence method as a PhD student when somebody came to my lab with about 1.7 milligrams of protein. I characterize the whole thing. What are the amino acids, N-terminus, what are the tryptic peptide, what are the N-terminus, and showed that it's a new kind of a protein. And, I gave them back 1 milligram of protein back. And I'm talking on 1978. So what I'm trying to say is that fluorescence gives you that advantage. And I remember I built my own amino acid also at that time using a reagent called fluoroscopy. So I could detect picomole to femtomole level, whereas ninhydrin needs nanomole levels of amino acid. So sensitivities are improved as you start moving from FDNB kind of molecule to fluorescent molecule. And I'm sure today at the end of the lecture, we'll be hearing Jagannath talking about how he's going to do it. Zeptomole, that is 10 to the power minus 21 mole, that is few hundred molecules. How can we do it? Yeah, we are going to hear that. So that's how it has moved from 1950s to 2020. So the strategy, as I said, he used performic acid. It will separate into two things. So you just separate them, elute them, and the rest is story. So the first person, Tapi, who came from Austria, he worked with him, and he worked on the A chain had amino terminus as glycine, and the second one, that is, which was called the B chain, had at the amino terminus phenylalanine. Okay. And they said, okay, we will work out on the B chain because amino acids were well distributed. There were average only one amino acid of one type in this chain. So there were not too many repeats. Unlike in A chain, there are some amino acids which are repeating very many times. So they said, okay, let us do with the B chain. And at that time, nobody ever had the guts to use enzymes to proteolytic enzymes, because if there is a fear that enzymatic reactions can do both catalyze the forward reaction as well as backward reaction. So the worry was that if a peptide bond breaks and the enzyme might join some other peptide and create another type of thing, and we'll be having a mess at it. But when they studied this, they found that nothing like that happened. So that was the first time they said, okay, I can use this trypsin to break it. So they got good fragment, and Tapi finished all the 30 amino acids I identified in less than 1 year working with Sanger. Okay. Another PhD student came from Australia called Ted O'Thompson, and he did the A chain for his PhD work. And you can see various samples. They took insulin to make sure that which amino acid. Here you can see the typical paper chromatogram showing up there. Okay. And so this is the handwriting of Fred Sanger himself. So they found that this is how the sequence is, but they didn't know where are the intramolecular disulfide bond and where is the intermolecular disulfide bond, except they guessed that probably this might be linked, nothing more they knew. So this was another challenge they had to solve, how the disulfide bonds are, and that took him another 4 years. Okay. So again, he used the performic acid, but this time he used the performic acid after breaking the peptide bond with trypsin or something, the insulin. He separated the peptide so the disulfide bonds were intact. Then after purifying it, then they subjected to performic acid. They will only get 2 peptides. So they had to just sequence that so they know this one is linked to this one. And this was done by Ryle, Smith, and Kitai. This is the last round experiment. And in and he came out with this beautiful structure. As you can see, how the within the molecule, within the A chain, there is a one this thing, whereas between A and B chain, there are 2 intermolecular between the molecule. So this was a great thing. So first time they showed amino acids have a particular sequence in which it is arranged. So sequence is very, very important. And number 2, they are all linked via alpha amino, alpha carboxyl. And that is the first time we established that proteins have a regular defined structure. Okay. Now you will see when I go on that, what is the relationship between the genetic material, what is DNA and protein, etc., were the questions which were asked later. Okay. In the meantime, VM Ingram used to work with Sanger, and he got a position in Caltech in Linus Pauling's lab. And Linus Pauling, one of the most brilliant minds we have come across, he discovered that when he did electrophoresis of cells, which were RBCs, from a sickle cell and a normal human being, he found they moved differently. So he had the brilliant idea way back in 1948, saying that, look, there must be a charge variation. So VM Ingram joined the lab and did what is called fingerprinting. What we do in fingerprinting is you take the paper, spot it on one end, do a paper chromatography, that is with butanol, acetic acid, water, and then after separating it, the separated one are subjected to electrophoresis because amino acids have a charge. You can separate them, and you can see the normal hemoglobin and sickle cell hemoglobin is on the right. All the peptides are same except one of the amino acid peptides has shifted, as you can see. This one is here, this one has moved over there. All the rest are identical. So you simply elute out that peptide, find out the amino acid composition. Immediately they discovered the amino acid replaced in the normal hemoglobin is valine, it is replaced by glutamic acid. And that is the first time we started learning that what happens, that a mutation leads to change in amino acid. And in 1958, it was a major sensational discovery. Okay. Of course, after that, the molecular biology, the new buzzword came, and so DNA double helix was proposed. Then, 1953, it was confirmed in 58. Then people had, interestingly, one of the Indian Anand Sarabhai, who was doing PhD in Cambridge, he demonstrated through a series of beautiful experiments that the DNA and the protein are related, what is called collinearity of the DNA and protein. So they said, how is the message in the DNA read into the protein? And so a word called messenger RNA was there must be some messenger RNA around, and there was a huge discovery. In the meantime, it was confirmed that what the model Watson and Crick had proposed, that it's a semi-conservative replication, was confirmed experimentally by using ultracentrifuge. Okay. Another thing they discovered, that they could do a cell-free translation of protein. So they could create protein with cell-free. You didn't need a whole cell. So that was something amazing people had developed. Another, as I said, collinearity of DNA and protein, which was PhD work of Anand Sarabhai, Vikram Sarabhai's nephew. Okay. Another very interesting observation which happened was somebody made polyuracil, put it into the cell-free translation, and he made polyphenylalanine. So they didn't knew that mRNA, that's why the word messenger RNA came and not messenger DNA. Okay. Then this led to the concept of DNA goes into mRNA to protein. Then, of course, you know the famous how the triplet code was deciphered, and Hargobind Khurana was one of the major players in that. And then another discovery came called restriction enzyme, and Sanger has been waiting for enzymes to cut DNA. So the discovery of restriction enzyme and DNA polymerase, when it happened, he got the tools for cutting DNA, and the rest is how he started sequencing DNA. Okay. This was also period when what we call as modern biotechnology took place, because recombinant DNA technology became popular, monoclonal antibodies became popular. All these things happened in the 70s. So people said, now as you see earlier, before this recombinant DNA technology, our major source of insulin was pig pancreas. And it has been estimated that for one man's requirement for 1 year, we need to have 106 pigs. And today you guess it is claimed we have 50 million people, so we'll need nearly 6 billion pigs. But thanks to modern biotechnology, we are able to make a bacteria which never made an insulin, make insulin, grow it in a fermenter, and then produce it. And then people have changed things. Now, earlier, when you used to have pig insulin being given, normally we had to give it 3 hours before we go for our lunch. So if you take insulin shot at 10, by 1 o'clock, you must have your lunch. Thanks to modern biotechnology, we have fast-acting insulin, slow-acting insulin, etc. And I don't want to go into details of that, but I just want you to know that we could create insulin. And one of the companies which became very famous was the Genentech, which went on to make the recombinant insulin and then commercialized, and Eli Lilly took the thing, etc.
Sir, yeah,
Yes.
I mean, another 2 minutes
Yeah.
or a few
Yeah,
minutes?
yeah. I'll just finish now.
Sure,
Okay.
yeah.
So I just wanted to quickly go through that. So in the 60s, Sanger left this protein business because he couldn't find the function of insulin, mechanism of action. And today also we don't know. But he was clever enough to look, this is beyond my capacity. So he said, okay, I'll stick to sequencing. So he used this polymerase to do what is called the, how to add a nucleotide or a subtract a nucleotide. That's why it is called plus and minus. Okay. And then normally our DNA polymerase have a editing function that if the wrong nucleotide is added, it will be removed and this one will be added. Okay. And so he used what is called the Klenow fragment, which doesn't have that editing possibility. Okay. And then he used a judicious mixture of radioactive dinucleotide triphosphate, 4 of them, etc., and which you will hear from Raja in great detail. So use a gel, read the ladder, and you come to know this. So this was the first paper he published in 1975, Sanger and Coulson, and this is a method. I won't go into details of it. I'm leaving it for my other panelists to speak about it. Okay. And then you have, today genomic revolution has happened so much. So Sanger did it in 75, Maxam and Gilbert in 77. Then Sanger sequencing went through a lot of revolution, which you're going to hear from them. So you will hear a lot of techniques are developed, and Raja himself has done a lot of this work. So he'll tell you personal account of his experience. And with the Human Genome Project, they discovered from the sequence what are the proteins present, and as you can see, they identified what is the storage protein, what is the receptor, what is the isomerase, which is a hormone, where is insulin sitting, etc. All came out of this whole thing. So this is a beautiful illustration of how many different types of proteins are produced, which is necessary for a normal function. And I just quickly wanted to, I'm sure we'll talk more about it. When you look at modern biology under the microscope, Leeuwenhoek first time saw the, with the hand lens microbe. And today I can say which is the microbe present from the DNA sequence. So between 1998 and now, number of, we know the sequences of so many organisms, so many things that it is unbelievable. So you can have what is in our gut, what are the microbiomes, etc. happens. Okay. Lastly, we have a very brilliant panel of specters. So I'm going to hear Professor Sanjeev Galande, who will talk about, he has been working on epigenetics and how he uses mass spectrometry, much more from him. He is one of our bright students. He was from our, if I'm not wrong, from the 3rd batch of biotech from Pune University, and also a Bhatnagar Award winner. Then, of course, Raja is, you'll enjoy hearing him. He was from our, 2nd batch when he joined. Then he moved over to Madurai Kamaraj University, went on to do his PhD, etc., and started a company. So he's our 1st generation entrepreneur. He and his wife are an excellent combination and made possible genomic revolution in India. And today we are going to hear Dr. Jagannath Swaminathan, youngest amongst all of us, and how he is doing fluorosequencing and landscaping what is emerging out of single molecular protein sequencing. Lastly, I just want to say Sanger was a very humble person. As he rightly said, unlike most of my scientific colleagues, I was not academically brilliant. I never won scholarships and would probably not have been able to attend Cambridge University because his parents were very rich, so they could afford him. And however, when it came to research, where experiments mattered most, he bet some of the brightest of minds in the world. So I remember, I had invited him to, when I was in England, to Imperial College, and he only gives 5 lectures in a year those days. So I went and booked a day back in January. It was, sir, you must come and give a lecture. And suddenly they announced in October the Nobel Prize to him. So I called him up, sir, will you be coming? He said, what? I am coming. I'll rehearse the Nobel lecture in front of you. Is that okay? So I'll come. So before going to Stockholm, I'll see you and then go and rehearse. So such a humble man I've ever come across. And we still are waiting for the day when we'll have a cure for diabetes. So there's a flame which is burning in front of Banting's house, the museum which it has become, and we hope the younger generations who are sitting here would have this thing. Thank you so much.
Thank you so much, sir, for this trip down the history lane. I mean, simple and elegant experiments led through a breakthrough like protein sequencing. And I still remember, huddling in the biochemistry lab, which you conducted, doing the dancyl chloride method of, that paper chromatography, and getting excited that the amino acid is moving. So I can't forget that. So, thank you so much. Okay, so if there are no questions, I can't see any in the chat box. But in the interest of time, would it be okay if people reserve their questions towards the end and we move on to the next panelist so that we can have all the questions towards the end? Then, yeah, okay. So it takes me. It's a great pleasure for me to introduce Professor Sanjeev Galande.
Stop sharing screen. Ganpa, stop sharing screen.
Oh, okay.
Superb.
Okay. Yes.
I've already stopped sharing.
Okay.
Yeah, you have stopped.
Yeah, I've stopped.
Okay. Can I
We are
mute
on gallery
people?
view.
I think yes, yeah. Okay. Yes. Right. So it's a great pleasure for me to introduce Professor Sanjeev Galande. He is one of the leading epigeneticists, not just in the country, but worldwide. Contributions from his lab have shed light into how higher order chromatin architecture affects gene expression. He's presently the Dean of Natural Sciences at Shiv Nada University, but this move was very recent, and before this, he was at IISER Pune as the Dean of Research and Development, as the professor in biology, and also the founder and of the Center of Excellence in Epigenetics. And having known Sanjeev as a collaborator and mentor, I can say he's one of the few epigeneticists who just not works with one model system, but several model systems, starting from yeast, Hydra to C. elegans, Drosophila, mouse, and cancer cells, and thus looks at the evolution of epigenetic mechanisms. For his contributions, academic contributions, he's been given the in one of the highest science award in India, the Shanti Swarup Bhatnagar Award in 2010, and the G.D. Birla Award in 2014. And without taking much time, I would now hand it over to Sanjeev, and I'm very excited to hear from you about protein sequencing. Over to you, Sanjeev.
Thank you, Bhavna, for a very, very kind introduction. And of course, it's a great pleasure to be part of this conference organized by Professor Kannan, and also to see some of the familiar faces, including Raja and many others, and some students. So what I'm going to do now is, because Professor Kannan told me that this is going to be mostly aimed at the students, I have basically lined up a few introductory slides which will give an insight into how proteomics has changed over the last few decades. The story that you just heard about sequencing, characterization of one protein and the structure, how it evolved over decades of efforts of large number of people, and finally made a very remarkable contribution. So that was the birth of the science of proteomics, and of course now it has evolved greatly. And I'm sure you'll hear in the final talk by Jagannath about various new ways by which one can really take the sensitivity to few molecules. What I'm going to do is give you some ideas about, talk about what is happening in the field in terms of various ways in which proteomics can actually add to the growing knowledge in the field of modern biology. So if I can share this presentation.